And thank you so much, Carl, for your introduction and also the opportunity for us to introduce our critical antigens for COVID-19 serological test development. First, I would like to very briefly to introduce some background of coronaviruses. Coronaviruses was identified in 1964, and these viruses are a group of viruses with a clone-like appearance. And those coronaviruses are positive sense, single stranded RNA virus consisting of the largest RNA genomes. And the host range of the coronavirus are usually the mammals and also birds for different group of coronavirus. And at the right side of the schematic diagram, you can see that there are different group of coronavirus, and those group of coronavirus with the discovery year are the coronavirus infect human beings. Among those 7 groups, those 3 groups cause severe symptoms are the MERS-CoV and the SARS-CoV, and also the COVID-19 in the current pandemic. And these slides, I'm going to very briefly to cover the application of serological test in COVID-19. And as you know, that serological test can detect the past infection after the onset of symptoms, and the antiviral specific antibodies can last from short period of time to long period of time, or sometimes even lifetime. And the serological tests of SARS-CoV-2 can complement the other clinical diagnosis and help tracking the systematic and recovered patients to support the epidemiological studies. And usually right now, as you know, that the serological tests of coronavirus 2 in the markets are mainly test the IgG and or IgM. There are some efforts have been made to test IgA and or IgG. And from the publications, you can say those 3 different antibodies have different patterns of response to the coronavirus infection. Usually IgA and IgM response more rapidly in the early stage of the infection. And the IgA usually responds later and lasts for longer time. And so the serological test kits usually, can test those different type of antibodies for the diagnosis. And from now on, I'm going to introduce some efforts from ACROBiosystems to help the serological diagnostic test development. And basically, we will introduce our products and our efforts from 4 different parts. The first one is we have been making efforts to design a trimeric spike extracellular domain full length of SARS-CoV-19 spike proteins. As you know that the spike protein has tumoric tumoric form natively, and the tumoric form is very important for the infection and also virus activities, and also very important for the neutralizing antibody development and vaccine development, and also very important for the serological test development. And based on the structure, ACROBiosystems have designed the extracellular domain expression constructs specifically with all those elements, and especially at the C terminus, adding the trimer motif of bacteriophage T4 fibrinogen, and to facilitate the trimerization, and also introduced some sites mutagenesis at the region, the domain of between the S1 and S2 subunits, to try to remove the foreign cleavage sites. So in order to get the fullness of extracellular domain, and also to produce a more stable tumoric formats spike protein. And we have well characterized the tumoric spike extracellular domain protein of the COVID-19 virus. And you can see from the slides we have SDS page data to show the tumoric product has the right size based on the evaluation on under the reducing condition on SDS page for the monomer. And this is the seq-Mars data to characterize the tumoric spike protein. And you can see this data also show the proper tumorization, and show the right estimated molecular weight as a tumor. And also this data shows that our product has very homogeneous tumoric particles via the negative stain electron microscopy. And combine those three data, you can see that the tumoric form of the spike extracellular domain protein have high quality with the right tumorization, and also with the very uniform tumor population. The population is higher than 80%, and the quality is much higher than some other vendors' products, which expressed in insect cells, and you know, their product basically with very high polymers, and the quality is not so good as ACROBiosystems products. And the second part, we're going to talk about a little bit of the glycosylation profiling. These slides actually is from the published data. And this literature comparing the spike protein expressed in insect cells, and also the spike protein expressed in HEC293 cells. From this gel, you can see the mammalian cells expressed spike protein has higher molecular weights, is implying that there will be a higher glycosylation, and these are the glycosylation profiling for those two different expression systems. On the top, you can see that the spike protein expressed in insect cells has different different number of the N-glycosides, and also different type of the N-glycoside. And the spike protein expressed in mammalian cells has more complex and also has higher number of glycosides. And also the spike protein expressed in mammalian cells are more close to the native protein expressions. And from this slide, you can also indirectly demonstrate that our spike extracellular protein are more close to the native version, because most of our spike protein are expressed in HEC293 cells. Our products should have a more close glycosylation profile to the native form. And these slides, we're going to tell you some of the efforts we have made to optimize the expression constructs for the spike RBD protein. Since RBD protein can be used for developing vaccine, therapeutic antibody, or the diagnostic tests. And based on those two RBD constructs from two different groups, one is from Mensane, and another one is from the Reagan Institute. And those two constructs actually contains very close region of the RBD, but there's some difference between those two RBD expression region, and also the type is different. We tested those constructs expressed protein RBD protein in ELISA, and you can say from this chart, the region RBD recombinant protein seems to be performing better compared to Monsanto's RBD, and give you higher bioactivities. And also we have some feedback from other customers. They also show that different expression region of the RBD can give you the different detection sensitivity in their test. So based on those information, we are planning to screen multiple expression constructs to optimize the expression region to get the best sensitivity and specificity for your serological kit development. And we have also developed 5 different RBD mutants products. Those are the 5 mutants. They have different mutants in the RBD region. And we have also characterized those mutants in our ELISA test and also SPR test. And you can see that among those 5 mutants, there are 3 mutants show better binding affinity compared to the wild type. The red one is the wild type. The others 3 are the mutants, and they have their binding curve left shifted. And also compared to the wild type, those 3 mutants also have shown the better binding affinity in SPR. And we have done further characterization of those with those RBD mutants for the antibodies. There are 4 antibodies actually isolated from the patients in China. These are the data from our collaborators. They have isolated 4 different antibodies from the Chinese patients of COVID-19, and we tested them against the RBD wild type and those are 5 RBD mutants we have. And you can say that 3 of the antibodies can bind with the wild type and some other mutants. But for these mutants, only 1 antibody can bind with these mutants. The others did not show the binding. So this implies that different mutants in the population in your specific region or country can only bind to the antibody developed against these specific mutants. When you develop serological test kits, we suggest that it's better to consider the actual mutants in the local patient population and use that actual mutants to develop your serological test method. And we can also provide the customized production of your interest mutants for your serological development, test development. And this one is actually the summary of our products, the SARS-CoV-2 antigen products for the serological test. We have developed the antigen of spike protein and S1 proteins, S protein RBD, and also nucleocapsid protein. And all these proteins, except all these proteins expressed in HEC293 cells, except the nucleocapsid protein are expressed in either HEC293 cells or E. coli cells. And all the proteins have different tags, either with H tag or human FC tag or mouse FC tag, or with H tag and AB tag for the biotinylation. And the turmeric S protein we have just introduced with well characterized properties is a very good antigen candidate for the serological test.
Sorry for interrupting, Dr. Peng, but you have 1 minute left. Thank you.
Yeah, actually, this is my last slide. Thank you very much. And the last one, I would thank you all for your time and attention, and also thank Dr. Yinan Jiang for his kind sharing of all the information. One more, that's the NAMA rifle. You are welcome to join our NAMA rifle. That's it, that's my slide. Thank you very much. Is there any question?
Dr. Peng, thank you very much